Bismillahirrahmanirrahim. Video 2 on the June 24 question paper 2-3. Bismillahirrahmanirrahim. Question number 5. Cyclin dependent kinases CDKs are enzymes that regulate the cell cycle. Cell signaling by ligands causes the activation of CDKs in target cells. Outline the main stages in the process of cell signaling by ligands that can cause specific responses in target cells. Cell secrete ligands. Then the ligand is transported through the blood in the circulatory system. Ligands bind to specific receptors. The word is receptors. You have to use this word, is underlined. Binding of the ligand to the receptor sets off reactions within the cell, which triggers a secondary messenger, or it sets off an enzyme cascade. You could have talked of cyclic AMP as well for secondary messengers. So cells secrete ligands which are transported through the blood. Then ligand binds to complementary receptors in the context of target cells, and then sets of reactions within the target cells. You could have talked of the reaction sets of an enzyme cascade. That would have been right as well. Or you should have said triggers a secondary messenger. Any of these terminologies was acceptable by the examiner. Part B of the question says the activity of CDKs is reduced by CDK inhibitors. Many of these inhibitors occur naturally in cells. Figure 5.1 is a diagram of a CDK inhibitor binding to a CDK molecule. So inhibitor CDK active site. State and explain how the CDK inhibitor in figure 5.1 prevents the activity of the CDK molecule. Naturally, as you can say, it's a non-competitive inhibitor because it's binding to a site other than the active site, or which is also called an allosteric site. Binding causes a change in the shape of the active site, and active site is no longer complementary to the substrate. Very basic, simple English, but very exact biological English. Now you say the shape of the enzyme changes. That's wrong. It's the shape of the active site changes. And active site is not functional. No, active site is no longer complementary to the substrate. So that is why students lose marks, because you don't write the correct terminology. So wordings binds to site other than the active site, causes change in shape of the active site no longer complementary to the substrate. Part C of the question says table 5.1 lists 3 different CDKs, their roles in the cell cycle and molecules that inhibit them. So we have CDK1 and the inhibitor is RO3306, regulates cell progression from G2 to mitosis. So now let's go through the cell cycle and talk about that. CDK2 regulates the process of S phase. This is P21CIP1. CDK4 regulates cell progression from G1 to the S phase. And this is PALBOCICLIB. Now with reference to table 5.1, state which CDK inhibitor is likely to result in a cell containing one chromatid per chromosome. You see, chromosome is one, but when it replicates, then it forms sister chromatids. Now it says which inhibitor is going to result in a cell containing one chromatid. So that means no DNA replication takes place. No DNA replication means that takes place in S phase of interphase. So we will say P2, this one is the one which is going to cause the damage, because it will stop DNA from replication. Or you can say DNA replication occurs in the S phase of interphase. So the inhibitor P21CIP1 and DNA replication occurs in S phase. So if that is going to be inhibited, then naturally the chromosome will not form the sister chromatid. With reference to table 5.1, state which CDK inhibitor is likely to result in a cell with a relatively high concentration of mitochondria and true chromatids per chromosome. High concentration of mitochondria and 2 chromatids per chromosome. The inhibitor would be RO3306. Now, why will it be this one? This is what we need to understand. We're going to talk about this one. Regulates cell progression from G2 to mitosis. You see, in G1, mitochondria have replicated. So instead of 100 mitochondria, now there are 200. S phase has taken place, chromosomes have, DNA replication has taken place. Now the cell cycle stops in G2. Now instead of 100 mitochondria, now there are 200 mitochondria per cell. So cell cycle stops in G2 after mitochondria have divided or increased in number. Because mitochondria divide or increase in number in G1 and G2 before mitosis. Now no mitosis is going to take place, so number of mitochondria will remain high. DNA replication will have occurred. Sister chromatids will have been formed in S phase, but the cell cannot carry out mitosis. So that is why in one cell now there will be more mitochondria as compared to another cell which has not undergone G1, S and G2 phases. So RO3306 inhibitor, but you won't get any mark for that unless you give me the explanation. Cell cycle stops in G2 after mitochondria have increased. Sister chromatids will have formed in S phase, but cell cannot carry out mitosis. Now let's come to the D part of the question, which is the last part of this question. Scientists have developed CDK inhibitors that are synthetic. Explain why CDK inhibitors can be used to treat cancerous tumors. Yeah, well, of course, cell cycle is stopped before mitosis occurs. What happens in cancer? It's uncontrolled cell division, uncontrolled mitosis. So it stops the uncontrolled cell division, so it stops increasing the tumor size. So cell cycle is stopped before mitosis and stops uncontrolled cell division, which means there is going to stop increasing the tumor size. Coming to the last question, which is question number 6. DNA and RNA are polynucleotides. Describe 3 ways in which the structure of mRNA differs from the structure of DNA. They're only talking of the mRNA. There are 3 types of RNA, please remember: mRNA, tRNA, and ribosomal RNA, which is rRNA. In each of your answers include information about structure of RNA and the structure of DNA. So structure, not function. Now, you can easily draw a line here, write mRNA here, and DNA here. Who says you can't do this? Usually students say, Miss, can we do this? Of course you can do this. Single-stranded. This is double-stranded. Well, of course, I'm not saying you just write the abbreviations. You can write the whole of it. So single-stranded and double-stranded. Then RNA base is not paired. It's a single strand, DNA base pairing present. Then ribose sugar, deoxyribose sugar. Then uracil, adenine, cytosine, and guanine. And here you have thymine in DNA. You have thymine, adenine, cytosine, and guanine. So only difference is uracil and thymine. Adenine, cytosine, and guanine are present in both. Then shorter nucleotides, longer nucleotides. So, I mean, you could do a table and you could do this like this, writing the heading. Of course, I've done it on a separate page. Single-stranded, double-stranded, RNA bases not paired, DNA bases are paired, ribose, deoxyribose. The bases are uracil, adenine, cytosine, and guanine. Of course, I've just given the letters, but you would, of course, write all of them. So, adenine, cytosine, and guanine are common to both, only the uracil and the thymine is different. Then, fewer nucleotides or shorter nucleotides and longer nucleotides. Part B of the question says scientists have synthesized 4 synthetic bases Z, P, S, and B. Z, P, S, and B. The base pairing of the synthetic bases are shown in figure 6.1. D is equal to site where bond forms with deoxyribose. So now they're going to ask you something in this. So we have Z, P. Now please remember Z is single ring, P is double ring, S is single ring, and B is double ring. And there are 3 hydrogen bonds. Cytosine and guanine have 3 hydrogen bonds. And cytosine and guanine have 3 hydrogen bonds. So, I mean, there is some clue here which you've got to figure out. State the letters that represent the 2 purine synthetic bases. Now, purines are what? Adenine and guanine. Purines are adenine and guanine. So it says state the letters. The letters are P and B. Let's figure that out. You can see here P is double ring and B is double ring. So P and B are double rings and these are purines, and that's what they asked you. State the letters that represent two purine synthetic bases in figure 6.1. Then part 2, last question, last part. State and explain which DNA base pair is most similar to the synthetic base pairs in figure 6.1. Now state and explain which DNA is similar to the synthetic base pairs. Now let's look at that. Now here what do we have? They're both C and G. So they're similar to C and G. Why? Because they're held together by 3 hydrogen bonds, same as the artificial base pairs. So I mean, they were asking you how are they similar to this. So when we look at it, it is C and G. AT has double bond, 2 hydrogen bonds. C and G have 3 hydrogen bonds. And I tell you to remember it. C is the third letter of the alphabet. So C and G have 3 hydrogen bonds held together by 3 hydrogen bonds. So video 2 is completed in this, and we will continue with the other papers, which are the paper 4s and the 5s in the upcoming videos. Thank you very much.